So biological introductions are a threat for the modern ecosystem. They can work to homogenize environments, as pictured kudzu here. They can also outcompete native species and displace native species, along with other ecosystem interactions. And the biological introductions can contribute to overall biodiversity change, and that biodiversity change is usually in the negative. Traditional studies of biological introductions tend to focus on ecology and demography of the introduced species. Some examples of this could be herbivory interactions, so herbivore release in the introduced environment, also mating system shifts that could extend Baker's law in the introduced environment. Also, traditional biological introduction studies have focused on the introduced populations alone, typically because of ease of study, and fail to look into the native populations and specifically the source of that introduction. If you want to get a good glimpse at the evolutionary processes surrounding an invasion, you need to be able to adequately identify the source and perform your phenotype or whatever you are looking into evolutionarily on that source population. There's been recent evidence that rapid evolution is essential for introduction success. One of the best examples of this is a marine copepod, which has successfully invaded freshwater environments over multiple lineages. And the reason for the success is due to the rapid evolution in response to that freshwater environment. Also, biological introductions can be used as a model to study microevolution. Here I am going to discuss the invasive red seaweed, Gracilaria vermiculophylla, pictured in the top left. Gracilaria is a successful invader originating from the Northwest Pacific around Japan, and the map shaded in black. And it has been introduced to the red regions along Europe, the east coast of the US, and the west coast of the US. Accompanying the introduction, Gracilaria has undergone an ecological shift as well. Pictured in the top is a typical native plant that undergoes sexual reproduction and settles onto hard substratum. And pictured in the bottom is from the introduced populations, which are typically found on intertidal mudflats. And those populations aren't undergoing sexual reproduction and propagate clonally. The introduced populations that are typically found on intertidal mudflats are in high stress environments. Because of tidal influence, they experience strong salinity changes and strong thermal changes. So posed here are the questions: are introduced populations more resistant to heat stress? And do introduced populations display altered heat shock protein gene expression profiles? So to answer this question, we sampled 41 populations across the native and introduced range. There has been previous work indicating that there was evolution in thermal tolerance in the introduced range, yet that study failed to sample the known source of the introduction. And here we have sampled the source of the introduction, so we know we can have a better idea of how evolution has functioned surrounding thermal tolerance in the introduction. So this was conducted for field collected material and common garden material. The field collected material was 7 days out of field before the thermal stress assay was conducted, and there was no death between collection and assay. Also, to remove previous environmental conditions, we used a common garden setup where individuals were housed in stable laboratory conditions for about 4 months. For the purpose of this talk, I'm only going to focus on 25 populations, 15 native Japanese populations, including the known source, 10 introduced East Coast populations, and from each population we collected 20 phalli, or a proxy for an individual, and that is pictured on the right. And from each individual, I removed 4 actively growing apical tips and placed those in seawater at 16 degrees Celsius for 24 hours. After 24 hours, I exposed a tip to 1 hour at 40 degrees Celsius, 2 hours at 40 degrees Celsius, or 4 hours at 40 degrees Celsius. These tips were then held at 16 degrees Celsius for 8 days. Survivorship was scored based on ordinal loss of photosynthetic pigment, or bleaching, which is a proxy for death. And along with this, I investigated 2 heat shock proteins, the expression of 2 heat shock proteins, HSP70 and HSP90, which are known to interact and function as molecular chaperones and work to stabilize cellular machinery under stressful conditions. And the expression of both of these HSPs are known to induce in response to a stressor, and in this case, thermal stress. They were quantified through RT-qPCR, following the Mike guidelines, and it's relative quantification relative to 18S, and all the data will be presented in a delta-delta CQ, presented as a fold change. So to orientate you to all of my figures, the native Japanese will be represented by black bars. The introduced Eastern USA populations will be represented by red bars. And the left panel will indicate field-collected material. The right panel will indicate the common garden material. Along the x-axis, we have hours spent at 40 degrees C, so either 1, 2, or 4 hours. And along the y for this figure is proportion bleached, or the proxy of death. So if you look as the hours of exposure increased from 1, 2, to 4, the proportion of individuals that died or that bleached increased. But there was a significantly higher proportion of Japanese native individuals that died when compared to the introduced East Coast populations. This was observed in the common garden and observed in the field collected and observed through the common garden. For HSP70 expression, again, we have the same schematic with the field collected panel on the left, common garden on the right, black indicating the native Japanese, red indicating Eastern US. And overall, as time of exposure increased, there was a significantly higher fold change in expression of HSP70 when the introduced were compared to the native. So the introduced populations are inducing HSP70 to a higher level when compared to the native Japanese populations. Again, held through the common garden from the field collected. For HSP90 expression, we saw similar trends. The native Japanese populations, again, did not induce HSP70 to the high of level as the introduced Eastern USA populations. And overall expression was higher in those introduced Eastern USA populations. So returning to my posed questions, are introduced populations more resistant to heat stress? It appears that the introduced populations are surviving heat stress at a higher rate. And because it was maintained through the common garden as a purpose to remove previous environmental conditions, it indicates that there's a genetic component to this change in survivorship. Additionally, my question posed, do introduced populations display altered HSP gene expression profiles? Again, the introduced populations were able to induce HSP70 and HSP90 to higher levels when compared to the native populations. Again, maintained through the common garden, indicating a genetic basis for that difference. So, taken overall, is there a genetic hint at evolution, microevolution accompanying and facilitating the introduction of this red seaweed through its invasive range? And for heat stress, yes, there appears that there is a genetic basis for microevolution facilitating survivorship of heat stress. Additionally, the HSP mechanism could explain this phenotype, because there is strong correlation between HSP and the ability to induce HSP and the ability to withstand a heat thermal stress. Along with thermal stress, our group looked into osmotic stress across the introduced and native range by exposing individuals to a low temperature, to a low salinity. And again, it seemed that there was a genetic basis where introduced populations were able to survive a low salinity at a higher rate than were the native populations. And a similar result was observed for cold stress. So the introduced populations were able to survive cold stress at a higher rate when compared to the native populations. There's been previous work indicating that the introduced populations have an increased herbivory resistance, that they are more, that they can survive burial better, so they can survive very dark conditions at a higher rate than do the native populations. And they have this high tolerance for desiccation, so they can survive being in the intertidal and undergoing drying out, and then they can still survive after they become dry. Overall, this points to the fact that microevolution can facilitate introduction success, and introduced species shouldn't be viewed as only static, URI-tolerant organisms, and they should be seen as having the capacity to respond to the introduced environment. So with that, I'd like to thank some people, my committee members, Dr. Alstrand, Dr. Courtney Murrin, a list of people that helped me in the field, that helped me in the lab, our international contributors, collaborators, and field help from all across the Northern Hemisphere. And my funding sources in SF, the College of Charleston, and the graduate program in marine biology. All right, thank you.